Good morning, students. Our today's topic in the subject of chemistry, paper number 4, that is alternate to practical. In short, it is called ATP chemistry. So our today's topic is criteria of the purity. So there are two criteria of the purity. So concentrate yourself. We are going to start. The purity of the substance can be determined by number one, the chromatography techniques, and number two, the melting and boiling points determination. So our today's topic in the criteria of the purity is chromatography techniques. So concentrate yourself. We are going to start now. So chromatography. Chromatography is a technique used to number one, identify a substance, number two, determine the purity of a substance, and number three, separate two or more substances in a mixture. Principles of the chromatography. Different substances have different solubilities in the same solvent. The more soluble the substance, the faster it will dissolve in the solvent. The more soluble substance will get carried along faster by the solvent and more further ahead of the less soluble solutes. So concentrate yourself, keep these points in your mind because these points will be applied in the experiment later. So the more soluble the substance, the faster it will dissolve in the solvent. Number two, the more soluble substance will get carried along faster by the solvent and move further ahead of the less soluble solutes. Paper chromatography is a simple chromatography technique employed in the laboratory to deal with the small quantities of the substances and for mixtures with a large number of the substances. So the two types of the paper chromatography are, number one is ascending type and number two is descending type. So the first figure is ascending type, in which the solvent will be placed in the bottom, and the second type is a descending type, in which the solvent will be placed in the top. So concentrate yourself now. Ascending type solvent moves up the chromatography paper. So look now, the solvent will move up the chromatography paper, and in descending type the sample, the solvent will move down the chromatography paper. Okay, figure below shows how developed chromatograms look alike. So one of the solute is the sample. So if the substance is pure, if the solute is pure, then there will be only one spot of the chromatogram. But if the sample, if the solute is impure, then it depends how many impurities are together. So the number of the spots, the number of the chromatograms will be placed, will be formed on the chromatography paper. So this is the final level of the solvent, that is solvent front, and this is the original spot of the sample. So, and there are three solutes in the sample and there is one solute in the sample. So this sample is pure and this sample is impure because there are three solutes in this sample. A pure substance will only produce one spot on the chromatogram. So this is the example of the pure substance, which because it is producing only one spot of the chromatogram, whereas the impure substance will produce more than one spot on the chromatogram. Each substance in the impure sample will appear as the spot of the chromatogram. So it means there are three substances in this impure sample. So that's why three spots has been formed. Paper chromatography can be used to separate and identify the various components in the black ink, comparing with the non-substances. So paper chromatography is used for two purposes. Number one is to separate. Okay, used to separate and identify the various components in the black ink. This is the one purpose of the paper chromatography. And the second purpose of the paper chromatography is a mixture of the amino acids comparing RF values. So here we are going through an example which will explain how separating and identifying the components in a sample of the black ink. So look now, cut a long strip of the filter paper of about 6 by 20 centimeter in size. Use a pencil to draw a line 2 centimeter from the one end of the paper, and use a capillary tube to place a drop of the black ink on the line and allow it to dry. Beside the spot of the black ink, place a spot of the red, blue, and yellow dye each, and allow the spots to dry. So it means there will be 4 spots of black ink, 1 spot of the black ink, and 1 spot of the red, blue, and yellow dye each. So in total, there will be 4 spots on the line, and then these spots will be allowed to dry. Lower the paper into a gas jar containing a solvent, that is distilled water, until the spots of the ink are just above the surface of the solvent. So look now, here is the figure. You will take a paper 6 by 20, and you will draw a line that is 2 centimeter from one end with the help of the pencil, and then you will put a spot of the black ink, that is X, on the line, and 1 spot each of red, blue, and yellow dye, and these spots will allow to dry, and lower the paper into the jar containing the distilled water in such a way that the spots are just above the surface of the water. So X is the sample of the black ink, R is the red dye, B is the blue dye, and Y is the yellow dye. Here is a beaker, here is the chromatography paper, and pencil line is here, that is 2 centimeter from one end, and here is water. So look now further, leave it to develop. Remove the paper when the solvents has almost reached to the top of the paper. So when the spots will be reached up to the top of the paper, we will remove the paper, allow the paper to dry. The developed chromatograms is shown below. So here are the chromatograms that are developed. So look now, the sample is having 2 spots, 2 chromatograms. Red is having 1, blue is having 1, and yellow is having 1. So the conclusion is black ink is not a pure substance because it has produced more than 1 chromatogram. So it means there are 2 solutes into the sample. Black ink is a mixture of these substances, a mixture of the blue and yellow dyes. So these spots will be like blue and yellow dyes. So it means it is a mixture of the blue and yellow dyes. So there are 2 solutes into the sample. This is not pure. Okay, the next example is in the paper chromatography is identifying a mixture of the amino acids. A mixture of the amino acids can be separated by using paper chromatography. Amino acids are colorless substances. To locate the spots of the amino acids on the chromatogram, a locating agent is used. Look how. The chromatogram is sprayed with a chemical called ninhydrin. The locating agent reacts with the components to form a colored compounds. A developed chromatogram is shown below. So here is the developed chromatogram. So here was the spot of the sample that was placed on the line, that is drawn with the help of the pencil 2 centimeter from one end. So here is the pencil line. So we have put 1 spot of the 1 drop of the sample onto the line with the help of the capillary tube. And here is the chromatograms that are produced, that is A. Its distance is A from the line. Here is B, its distance is B from the line, and here is C, its distance is small c from the line. So this is solvent front. Okay, RF values are component A is equal to A over Y. So here is the distance between the solvent front and the pencil line. So this is the total distance. and this is the distance of A, this is the distance of B, and this is the distance of C. So RF values of A is equal to A over Y, RF value of B is B over Y, and RF value of C is C over Y. So look further now. The amino acids can be identified by its retardation factor RF values. RF is equal to distance traveled by the component over distance traveled by the solvent. So each substance has its characteristic RF value. Component A's RF is A over Y, of B is B over Y, and of C is C over Y. So by comparing calculated RF values with those found in the data book. So we have our data book in the lab in which some RF values are given of the different substances. So we will compare these values with the values given in the data book, and we will judge which substance and which solute is this one. So by comparing calculated RF values with those found in the data book, the identity of the substance can be found. So this was the lecture, students. So I hope so this will be much more easier and interesting for you to understand. So read this lecture again, understand it, learn it, and make your notes. Thank you so much.